Thanks very much for the introduction, Alina, and thanks to the Physiological Society for this opportunity to speak at Future Physiology 2020. I'm hoping this talk will prove to be last but not necessarily least in the programme. My presentation is focused on the impact of diet on climate change and physiological health, with a focus on muscle physiology in an ageing population. And I've also included an interactive poll, so look out for Rosie flashing up different questions throughout the slideshow. By way of introduction, Rosie from the Physiological Society has asked me to say a brief word on career advice, given that early career is the target audience for this conference. And my main piece of advice relates to the importance of mastering as many state-of-the-art methods or techniques in physiology during your early career as possible. I was lucky enough to learn from some of the best technicians in stable isotope methodology in the UK, in the Netherlands with Luc van Loon, and in the US with Bob Wolf for the measurement of muscle protein metabolism. And the next three slides demonstrate a typical laboratory setup with participants being intravenously infused with a labeled isotope. So on the right-hand side of the slide here, in this case, labeled phenylalanine, 13 carbon phenylalanine. Blood samples on the left-hand side of the slide being collected from the forearm vein, which is heated to arterialize the blood. And also muscle biopsies being collected from the vastus lateralis muscle on the left-hand side, and on the right-hand side of the slide, you see the actual muscle tissue. Our studies typically combine nutrition and exercise and/or physical activity, and have recruited participants across the life and health span. So the message to you as an early career researcher is to arm yourself with as many techniques as possible during your early career, because it becomes much harder to find the time to pick up these techniques as you transition into your mid-career. And I also know that the Physiological Society run techniques and based workshops, for example, Western blotting and PCR, that you can undertake. So in terms of organising this existing presentation, I've split the talk into three main sections. First, I'd like to introduce the problem or issue by contextualising the link between food choices, greenhouse gas emissions, and mitigating climate change. And in doing so, I'd like to highlight the proposed solution from a sustainability perspective per se. Second, and aligned with the remit given to me by the Physiological Society, I'd like to pose the question, what impact could dietary changes based on greenhouse gas emissions have for healthy musculoskeletal aging, and why? And I'd like to provide some insights from scientific data that we and others have generated using tracer methodology to compare the response of muscle protein synthesis, so the primary physiological regulator of muscle mass, at least in healthy individuals, to ingesting different animal and plant-based protein sources. And finally, I'd like to make some recommendations on where next for scientific research in terms of devising dietary recommendations, specifically protein nutrition, that encompass both improving human health throughout the lifespan and also preventing climate change. And I'll be highlighting the importance of multidisciplinary approach to solving this problem that includes human physiology alongside mathematical modelling. So in terms of section one, we know that climate change results, at least in part, from the emission of greenhouse gas emissions into the atmosphere, which causes global warming. And according to 2018 UK greenhouse gas emission final figures, diet accounts for between 20 and 30% of the UK's greenhouse gas emissions. And by extension plays an important role in climate change, with direct contributions from the impact of agriculture on greenhouse gas emissions, shown in green on the pie chart, particularly methane and nitrous oxide, and indirect impacts from the contributions of transportation, energy supply, and waste management that are denoted with the asterisks on the chart. This significant impact of diet on climate change occurs because greenhouse gas emissions occur at every stage of the food chain, as shown on this diagram. So from agricultural production on the left-hand side of the slide, so in the green box, bottom left, which relates to various farming methods and land use, through to processing and manufacturing in the yellow box, which includes packaging, retail, where transportation adds to diet-related greenhouse gas emissions, and also consumer activities in terms of storage and cooking, and finally the disposal of waste food. This pie chart illustrates the percentage contributions to total diet-related greenhouse gas emissions from different food groups based on UK data from the National Diet and Nutrition Survey in 2001. And given these data, most focus regarding diet in terms of reducing greenhouse gas emissions and mitigating climate change has centered around reducing dietary intakes of meat, primarily red meat and processed meat, in the red segment of the pie chart, and also dairy, as shown in the blue segment of the pie chart. And of course, this change in dietary pattern will have significant nutritional impact on patterns of dietary protein intake in the UK population and beyond, given that 60% of protein is derived from animal proteins, of which again meat and dairy in the red and white portions of these two pie charts respectively make significant contributions, as evidenced by the latest UK NDNS data. And this is true both for younger adults on the left-hand side, the pie chart on the left-hand side, and also older adults on the right-hand side, 65 years plus. So my remit for this Physiological Society talk was to focus on human physiological responses to a change in dietary protein source. And with this in mind, I'm going to focus on healthy aging and specifically on comparing protein from meat, dairy, and plant-based protein sources for the metabolic health of aging muscles, as the title suggests. So what impact could dietary changes based on greenhouse gas emissions only have for healthy musculoskeletal aging, and why? Well, it is well established that the natural biological aging process is associated with a gradual loss of skeletal muscle mass, which is termed myopenia, and strength, which is termed dynapenia, and collectively known as sarcopenia. And if we focus on the green line, whereas muscle mass typically peaks in early adulthood, between 20 and 30 years of age, the onset of muscle loss typically begins by the 4th decade of life, continuing at a rate of approximately 1% per year until the age of 70 years, and increasing to more like 1.5% per year over the age of 80. And this trajectory may be exacerbated by periods of muscle disuse, ranging from short-term limb immobilization due to a fall, to longer-term bed rest or hospitalization in a best bed rest or surgery kind of scenario. And once a critical level of muscle loss is reached, often referred to as this threshold of disability, the likelihood of multiple detrimental health-related consequences dramatically increases, including a reduced mobility and loss of independence, increased risk of falls, and increased risk of bone fractures. So from a metabolic standpoint, two possible metabolic drivers of muscle loss or muscle atrophy exist. The first is an increase in the rate at which muscle protein, shown here on the, so here's a muscle and here are muscle proteins, but the rate at which they are broken down back into their amino acid precursors. Or second, a decrease in the rate at which amino acids, either from the blood or the intracellular of the muscle itself, are synthesized into muscle protein, which we call muscle protein synthesis, and which we know is controlled at the molecular level by mTORC1 signaling cascade, so the mechanistic target of rapamycin complex 1 signaling cascade. But of these two processes, the general consensus in otherwise healthy adults is that an impaired response of this process, i.e. muscle protein synthesis, is primarily in response to food intake and physical activity, is primarily responsible for the age-related loss of skeletal muscle mass. And this concept has been coined the age-related muscle anabolic resistance. As a result, a key focus of research studies in this field has been focused on characterizing the response of muscle protein synthesis to different protein sources in older adults, in essence with a view to overcoming this age-related muscle anabolic resistance, which leads to muscle atrophy. In terms of optimizing the protein source for the stimulation of muscle protein synthesis in older adults, it's currently understood that the interaction of 3 factors determines the potential of a protein source to stimulate muscle protein synthesis. First, in the red circle, is the digestibility of the protein source determines the bioavailability of amino acids as substrate for the synthesis of new muscle protein. Second, in blue, the protein source must constitute a complete profile of all 9 essential amino acids, of which a high leucine content in the green circle is necessary for the maximal stimulation of muscle protein synthesis. And this brick wall or building site analogy nicely illustrates how important the essential amino acid profile and leucine content of the protein source are for stimulating muscle protein synthesis. So muscle protein may be thought of as the brick wall on the right-hand side of this slide, and amino acids as the individual building blocks on the left-hand side of the slide, which are necessary to build the wall. And each type of wall requires a particular arrangement of these bricks. Certain types of bricks are always, so primarily the non-essential amino acids are always lying about and available endogenously at the building site. However, the wall can only be completed if other bricks, namely a full complement of essential amino acids derived from a dietary protein source, are transported in from outside the building site. The mTOR signaling pathway may be thought of as the builder that puts the bricks together to form the wall. Leucine may be considered to be a special brick. It's necessary as part of the wall, but it also comes with a pot of coffee that can stimulate the worker into faster and more effective wall building. So if the truck delivers a protein source constituting a full complement of essential amino acids, including a plentiful supply of the red brick leucine, the brick wall will be built effectively and efficiently. This slide summarizes characteristic differences in the essential amino acid profile and leucine content between protein sources, highlighting the differences between animal-based protein sources on the left in white and red blocks, and plant-based protein sources on the right-hand side in green. Sorry, the animal proteins are on the left-hand side, plant proteins on the right-hand side. And as you can see, the total essential amino acid content, so on the y-axis, of animal-based proteins typically exceeds plant-based proteins. And indeed, whey, milk, and casein are the only protein sources with a higher constituent essential amino acid composition compared with human muscle itself. Moreover, animal proteins, animal-based protein sources are typically boast a complete essential amino acid profile, i.e. containing all 9 essential amino acids, whereas most plant-based protein sources are deficient in 1 or more of these essential amino acids, and usually that's either lysine or methionine, as denoted by the black font on this slide. As a general rule, the leucine content of animal-based protein sources is typically greater than plant-based protein sources, although there are some exceptions to this rule. So while it's clear that animal and plant-based protein sources differ in terms of their constituent amino acid profile, the key question is, what's the difference between protein sources in terms of their anabolic capacity in the context of preserving muscle mass with advancing age? So in terms of experimental laboratory controlled studies that have directly compared the capacity for plant and animal protein sources to stimulate muscle protein synthesis, the most common comparison is between the dairy proteins and soy. And in this study by Sarah Wilkinson from Stuart Phillips' lab in Canada, they took a group of resistance-trained young men who consumed either fluid skimmed milk or a soy protein beverage immediately following an acute bout of lower limb resistance exercise. And both drinks contained a meal-like 18-gram dose of protein and were matched for energy content. The ingestion of milk protein was shown to stimulate a 34% greater increase in mixed muscle protein synthesis rates during exercise recovery compared to the soy protein condition. And this greater anabolic response to the milk compared to the soy was likely mediated by several factors, as shown on the clipboard here, namely the greater total protein digestibility, the superior essential amino acid profile, and leucine content of the milk. And there is evidence as a logical follow-up to this that the whey component of milk is primarily responsible for the greater anabolic response to milk ingestion compared with soy. And this time, these studies were conducted in older adults. So this first study on the left-hand side demonstrated that the ingestion of 20 grams of whey protein stimulated a greater response of muscle protein synthesis, both under resting conditions on the left-hand side of the figure, on the left-hand side, and in response to exercise in healthy older adults when directly compared to a matched dose of mycellar casein in the hatched bars. And studies that characterize the dose response of muscle protein synthesis to ingesting whey protein, so this is now the figure on the right-hand side, or soy protein under both rested and post-exercise conditions, also demonstrate a greater stimulation of muscle protein synthesis in response to whey versus soy protein at each protein dose. And again, this was in older adults. And apologies that the asterisks actually moved, so they should be above the 20 and the 40 gram doses. Two other studies provide a direct comparison between animal and plant-based proteins in terms of stimulating muscle protein synthesis in older adults. This first slide shows data from an unpublished study, again from Stu Phillips' lab, which compared the acute postprandial response of myofibrillar protein synthesis. So these are the muscle proteins, actin, myosin, tropomyosin, that are responsible for changes in muscle mass, the contractile proteins. And they measured postprandial protein synthesis rates in response to ingesting either 4 ounces of lean beefsteak versus an isonitrogenous soy protein-based beef replacement under both resting and post-exercise conditions. And this was performed in middle-aged men. And the key finding from this study was that beef ingestion elicited a greater stimulation of muscle protein synthesis, both at rest and following resistance exercise, compared to the soy protein ingestion condition. And this recent study compared the response of protein synthesis rates under just resting conditions to the ingestion of mycellar casein in the red versus wheat ingestion. And this, again, was in a cohort of older adults under resting conditions. And wheat protein was chosen in this study since wheat is the most abundant plant-based protein source in our diet, comprising approximately 25% of total protein intake, although that does depend on region and country-specific levels. And these data demonstrate that the ingestion of mycellar casein protein stimulates a greater resting response of myofibrillar protein synthesis compared to the dose-matched quantity of either whole wheat or the wheat hydrolysate, in which the anti-nutritional factors were removed, such that the total digestibility was similar to the casein. And this greater stimulation of muscle protein synthesis with casein compared to wheat, again, was likely attributed to the more favourable essential amino acid profile and leucine content, since wheat protein is deficient in the essential amino acid lysine, and the constituent leucine content of casein is higher than wheat protein. So if we take these different experimental studies together, based on our existing knowledge, based on the current studies that are available to us, the general consensus is that the ingestion of animal-based proteins, in particular the dairy proteins, are more potent in terms of stimulating muscle protein synthesis during exercise recovery compared with plant-based proteins. However, as an important caveat to this point, the strength of this statement is limited to the comparison with only 2 plant-based proteins, namely soy and wheat. And of course, there's much more work that needs to be done in terms of comparing with other plant-based protein sources. And of course, from the standpoint of environmental sustainability and food security, plant-based protein sources may be considered advantageous over animal-based protein-rich foods. And this figure quite crudely classifies estimates of greenhouse gas emissions associated with the UK's supply and production of different food groups into three categories, namely low, medium, and high. And as you can clearly see from the figure, plant-based protein sources fall into, in the green, fall into the low and moderate category, whereas animal-based protein sources essentially fall into the moderate and high category in terms of greenhouse gas emissions. So where next for the study into the anabolic properties of plant-based proteins? Well, based on these recent analysis of the essential amino acid and leucine content of different isolated proteins that this was a study that, it was from Luc van Loon's lab that used the same methodology to determine the essential amino acid or amino acid profile of these different protein sources. It appears that based on this analysis, that corn and potato protein sources and also pea protein were particularly relevant in terms of essential amino acid and leucine profile. And indeed, this recent study, again from Stuart Phillips' lab that was actually published just a few months ago, albeit in young women, demonstrated the capacity of potato protein to stimulate rates of muscle protein synthesis, both under rested and exercise conditions. But unfortunately, no comparison to an animal protein source was included in this study. Moreover, if we base things on rate-limiting essential amino acids, combinations of different plant proteins can be identified. So if we combine the lysine and the methionine content of different plant-based protein sources, and to orientate you, the red, if the circles are the same color, they suggest complementary plant-based protein sources. So based on this, it seems that combinations of potato and rice protein, corn and pea protein, and soy and hemp protein appear to be complementary in terms of providing a complete essential amino acid profile for supporting rates of muscle protein synthesis in older adults. And if we translate this science into practice, here are a few different meal plans that could be provided to cover all of these complementary amino acid sources. And of course, at the risk of opening a can of worms, a similar gap in knowledge also exists for insect-based sources of protein. And not to put you off your breakfast, lunch or evening meal, depending on where you are in the world, based on amino acid profile, beetles, ants, and termites are promising protein sources for future study in the context of stimulating muscle protein synthesis rates. So finally, where next for scientific research into devising dietary recommendations, and specifically with regards to protein nutrition that encompass both improving human health from a musculoskeletal perspective throughout the lifespan, but also preventing climate change? Well, I think it's clear that a multidisciplinary approach is required, where we combine our knowledge of nutrition and physiology with a mathematical modelling approach. And as one example, this study utilised a technique called linear programming to model the level of dietary changes, which are shown here. So to orientate you, the red bars are the current diet in this group of UK individuals, and the white is what was modelled to be a sustainable diet. And so what's projected to be a sustainable diet. And the key point here is that the changes in terms of the sustainable diet are based on attracting a diet that would match the requirements for health based on NDNS recommendations, but would also meet the target reductions in greenhouse gas emissions via dietary means, from government-based dietary means. And as you can see, a balanced rather than an extreme approach appears to be possible, i.e. a sustainable and healthy diet can be achieved without completely eliminating meat and dairy from the UK diet. And I think we will see more and more data out that model existing knowledge regarding human physiology outcomes, such as muscle health, muscle mass, muscle strength, nutrition, and sustainability, with a view to devising recommendations that account for both health and sustainability. So to summarise, I hope I've convinced you that diet makes a significant contribution to global warming and climate change. Second, that the age-related decline in skeletal muscle mass is mediated, at least in part, by this concept of muscle anabolic resistance. The next take-home message from this presentation is the anabolic potential of a protein source is dictated by three key factors, namely its digestibility, its amino acid composition, in particular its leucine content. On the basis of current scientific knowledge, I think it's fair to say that animal-based protein sources are more anabolic than plant-based protein sources, at least on a dose-match basis. However, I would reiterate that caveat that that's based on our current knowledge in comparison to only 2 plant-based proteins. And finally, moving forward, future protein recommendations should, in my view and others, should take a holistic approach by considering muscle health, sustainability, appetite, and other nutrients of concern that likely involved a marked increase in plant-based foods without the elimination of meat or dairy. I'd like to finish by thanking everybody that helped put this presentation together, all of those different people that I've worked with on these types of studies. Thank you all for listening as an audience, and I'd welcome any questions that you do have. Thank you.
Thank you, Ali. That was a really nice talk. We have questions streaming in here, so we'll just get started. The first question is from Ed Batty at King's College. Can you provide some insight into the biological significance of a 0.05% per hour difference in muscle protein synthesis? I.e., to what extent do you think this magnitude of difference in protein synthesis elicited in response to soy versus animal protein would influence the ability to maintain, build, or prevent losses in muscle mass?
Thanks for the question, Ed, and I can just be clear, this wasn't planned, even though Ed and I are at the same institution. Yeah, great question. So the answer to the question relates to, do changes in muscle protein synthesis translate to changes in muscle mass? And that's a really good question, and it's been a question that's been addressed in a number of different studies. Has been potentially a quite controversial topic, but my response would be quantitatively, perhaps not, in that if you say get a 0.02 or 0.03 greater percentage per hour increase with say whey compared to another protein source, that from a quantitative perspective, that doesn't necessarily translate into a greater increase in muscle mass or a greater preservation. But from a qualitative perspective, I think it's fairly safe to say that there is a link, that there is a translation. So, so to summarize, if you are getting a greater response of muscle protein synthesis with one protein source, you can assume that that response is going in the right direction qualitatively in terms of muscle mass, but not necessarily the quantity of that increase is the same. So hopefully I've answered your question there, Ed.
Thank you. We have now an anonymous question where they asked, when talking about sustainable diet, does that take into account sustainability of production, such as for ocean fish? I guess, I mean, was it purely CO2 in the calculation, or was it sustainability and ecologically?
Yeah, that's a great question. And it really comes down to how complex sustainability is. So I think, as the question kind of alludes to, in terms of determining the greenhouse gas emissions, so the environmental impact of different foods, then there are a number of additional factors that link in to determine how big an impact in terms of carbon footprint that food source will have. So the slide that I put up was very crude, and hopefully I did mention that in the presentation, in that they were kind of just guesstimates for these different food sources to give an idea of how different foods can potentially impact carbon footprint. But I do take on board the point that there's a lot of sub-factors that play an important role, i.e. where it's sourced, the location of where that food is sourced, whether it is transported or whether it's consumed in the country in which that product was produced. So I think it's a really important point. But my answer is that sustainability is incredibly complex, particularly when you even think even larger scale in terms of sustainability is not only about environment, it's also about the economy. There's no point recommending a healthy, environmentally sustainable diet if it's going to cost 3 or 4 times more per week for the average family. So sustainability is really complex, and hopefully that answers that question to a degree.
Thank you. We have another one from another anonymous attendee. What do you think about meat replacement products like the, oops, they're moving around, like the Impossible Burger, which tries to simulate the taste of meat, but is purely plant-based? And I can add to that, I mean, during your talk, I was also thinking about like hemp milk and almond milk and stuff.
Yeah, really topical question. I've been at a couple of recent industry conferences whereby we've been able to actually taste some of these alternative sources, and from a personal perspective, I found them very palatable, enjoyed them, and yeah, I think from a accessibility perspective, as in, you know, people actually consuming them, I personally don't see any issue with that. I'm not trying to advocate a plant-based burger over a meat-based burger. However, from a personal perspective, for me, it was palatable. So I think, again, more research will come out in terms of the biological value or the effectiveness of these different protein sources.
We have a question from JP Desforges at McGill University. Do you have any idea about acclimation, i.e. do long-term vegetarians utilize plant proteins to a greater extent than meat eaters? Can this influence the results of the presented studies?
Yeah, really tough question to answer in terms of, to my knowledge, I don't think that there's data available that directly answers that question. However, I think the point's a really good one in terms of adaptation to a diet. So I think there is lots of evidence out there that it can take a period of time to adapt to a certain diet, and from a metabolic standpoint, that does make sense. So it is a good point when you're interpreting these types of studies in terms of are these already vegetarian individuals? Are they not vegetarian, but they're changing to a vegetarian diet? So there are considerations when designing these types of studies, but to my knowledge, no study has systematically looked at that in the context of muscle protein metabolism in older adults. So, but yeah, something that's worth looking into in the future, I'm sure.
Yeah, we have a number of questions. We don't have time for all of them, but we could take 1 or 2 more. This one is an anonymous question. Has there been any research into protein content from lab-grown cell-cultured meats? Or would cell-cultured meats be able to have essential amino acids added?
So not in the context of these kind of controlled laboratory studies that measure muscle protein synthesis rates. There haven't been any studies yet. That's not to say that those types of studies won't be coming out in the future. Similarly, studies with looking at different plant-based proteins, insect protein, I feel that those studies are probably not too far away. So definitely keep an eye on the literature in that context. And in terms of adding essential amino acids or adding, for example, leucine to these types of products, yeah, that's absolutely possible. And there are a number of studies in this field that very much look at the impact of enriching products or food sources or isolated proteins that are perhaps deficient in some amino acids with additional essential amino acids. So, so again, that is an area that is ripe for research.